All living organisms, whether plants, animals or microorganisms, contain innumerable organic compounds or biomolecules in a certain concentration. This concentration is expressed as moles per cell or moles per liter. Interestingly, biomolecules constantly break up and are remade, or in other words, they have a turnover. They change into other biomolecules or are made from other biomolecules due to constant chemical reactions in living beings. The entire set of biochemical reactions that takes place in living organisms and helps them maintain life is known as metabolism. Metabolism is important to living beings to maintain their structures, growth and reproduction, and helps them respond to their environments. However, metabolic reactions lead to the transformation of biomolecules. For example, the decarboxylation of histidine gives histamine. Similarly, the hydrolysis of a glycosidic bond in sucrose yields glucose and fructose. Most of these reactions do not occur in isolation and are usually linked to other reactions. In fact, the series of linked chemical reactions that transforms biomolecules is called metabolic pathways. These pathways take a specific path and include specific biomolecules. They can be either linear, as in the process of glycolysis, or circular, as in the Krebs cycle. Metabolic pathways can be divided into two categories: anabolic pathways and catabolic pathways. Anabolic or biosynthetic pathways lead to more complex structures from a simple one. For example, the process of glycogenesis, which involves the biosynthesis of glycogen from glucose. In other words, glucose molecules are added to chains of glycogen for storage. On the other hand, catabolic pathways lead to a simple structure from a complex one. They involve degradation and also lead to a release of energy. For example, the process of glycogenolysis, which leads to the breakdown of glycogen to glucose. The energy released during degradation is trapped by living organisms and stored as chemical bonds. Later, this bond energy is utilized for biosynthetic, osmotic, and mechanical work whenever the need arises. The most important biomolecule that contains energy in its chemical bonds is adenosine triphosphate, or ATP. When bonds of ATP break, a lot of energy is released. ATP also transports chemical energy within cells for metabolism. All the chemical reactions associated with metabolic pathways are catalyzed reactions. Metabolic pathways can be compared to automobile traffic, where they have traffic junctions when they meet and cross each other. Just like automobile traffic, the flow of metabolites through these pathways is definite and takes place in a specific direction. This flow of metabolites is also called the dynamic state of body constituents. As you have studied, thousands of metabolites or biomolecules are present in living organisms. Each of these biomolecules has a definite concentration. For example, the blood concentration of glucose in a normal healthy individual is 4.5 to 5.0 millimolars, while that of hormones is measured in nanograms and milliliters. Moreover, these biomolecules are constantly a part of the metabolic process. Any chemical or physical process moves naturally to equilibrium. However, systems at equilibrium cannot perform work. Therefore, the living state is a steady state or a non-equilibrium state, which is essential for living organisms to be able to work constantly. The living state or non-equilibrium state is attained by energy input through metabolism. Hence, metabolism and the living state are synonymous, and one cannot exist in the absence of the other. Did you know that cheese is produced with the help of rennet, which is a natural complex of many enzymes? Enzymes act as biological catalysts in a living system and change the rate of reactions without themselves getting altered. While most enzymes are proteins, some nucleic acids also behave like enzymes. These nucleic acids are called ribozymes. Just as with proteins, enzymes have a primary, secondary, and tertiary structure. While the primary structure is an amino acid sequence, the tertiary structure folds and crisscrosses on itself, creating many crevices or pockets. One of these pockets is called the active site. A substrate fits into this active site of the enzyme and enables it to catalyze reactions at a high rate. While enzymes catalyze chemical reactions, they are very different from inorganic catalysts. Inorganic catalysts such as platinum and nickel work effectively at high temperatures and under high pressure, while enzymes are damaged at temperatures above 40 degrees centigrade. However, some enzymes, such as thermolysin, can be isolated from thermophilic organisms that live in high-temperature environments like sulfur springs. These enzymes retain their catalytic power even at temperatures as high as 80 to 90 degrees centigrade. To understand the working of enzymes, we'll begin by learning more about chemical reactions. Chemical compounds undergo physical and chemical changes. When a compound changes shape without breaking its existing bonds, it is said to be a physical change. For example, ice melting to form water. On the other hand, when the bonds in a compound are broken and new bonds are formed during transformation, it is called a chemical reaction. The rate of a reaction is defined as the amount of a product formed per unit time. It can be expressed as delta P upon delta T. Various factors such as temperature and pressure influence the rate of reaction. This rate generally doubles or decreases by half for every 10-degree change in temperature in either direction. For example, consider that a particular reaction is taking place at 5 degrees with the rate of reaction as 1 unit. When the temperature is increased by 10 degrees, the reaction rate will double to 2 units. Similarly, if the temperature is decreased by 10 degrees, the reaction rate will become half. A catalyst greatly influences the rate of a reaction. Hence, the rate of enzyme-catalyzed reactions is significantly higher than uncatalyzed reactions. Consider the example of a reaction where carbon dioxide and water react to form carbonic acid. In the absence of any enzyme, 200 molecules of carbonic acid are formed in an hour. However, when the enzyme carbonic anhydrase is used, 600,000 molecules of carbonic acid are formed every second. Hence, the carbonic anhydrase enzyme accelerates the reaction 10 million times. There are numerous types of enzymes and each of them catalyzes a unique chemical and metabolic reaction. There are also multi-step reactions, where each step is catalyzed by a single or multiple enzymes. For example, glucose is converted into pyruvic acid through 10 different enzyme-catalyzed metabolic reactions. Pyruvic acid yields different types of end products during various pathways. During aerobic respiration, it forms acetyl-CoA and carbon dioxide in the presence of the pyruvate dehydrogenase enzyme. While during anaerobic respiration in the muscles, in the presence of the enzyme lactate dehydrogenase, pyruvic acid leads to the formation of lactic acid. On anaerobic respiration in yeast, the same pyruvic acid leads to alcoholic fermentation in the presence of the enzyme pyruvic decarboxylase.